So let's talk a little bit about microbial communities and how they kind of live and interact together. We're going to talk about a specific structure which is referred to as a biofilm. And a biofilm is kind of a way that different bacteria who can act together kind of live together. So a biofilm is kind of a community of different types of organisms. And these organisms are joined together through different structures that are very sticky in a sense. So they maintain something which is called a glycocalyx. You don't need to know that for your upcoming exam, but the glycocalyx helps to provide kind of a protective layer and enables kind of the two different types of bacteria to form a meshwork with each other. And it doesn't have to be just two, it could be 2, 3, 4, 5, millions of different types of bacteria that can produce these biofilms. But they are complex in the sense that we don't fully really understand how they're woven together, because they are a giant creation of different, or a giant living arrangement of different organisms. So a key example of a biofilm would be that of dental plaque, which I know you're all familiar with because you're excited to clean that once you get out of this course and once you graduate from York Tech. But these types of biofilms are real important, especially in a clinical setting, because if you don't know about them, they can lead to nosocomial infections. So there's a lot of medical instruments that can harbor biofilms, or they make a good environment for a biofilm to grow. One example would be that of a catheter. And the catheter that isn't cleaned properly, or is cleaned but not thoroughly cleaned, it can create a nice environment for biofilms to develop. So streptococcus, staphylococcus organisms can reside in these types of locations, or on these types of instruments, because they haven't been thoroughly deconned and are being used before that takes place. So biofilms, as I said, they make up a variety of different species, and they are surrounded in a very sticky kind of matrix, which consists of many different polysaccharides being expressed by the bacteria. And so the bacteria will grow in very small clusters referred to as microcolonies, and they also are hydrated by what's referred to as water channels. So in a sense, there you have this giant community kind of working together to make sure that nourishment is provided to all cells, so that way the entire colony, or this entire biofilm, is thriving. Biofilms, like I said, are, they're pretty important in the clinical setting. Urinary catheters is one example, but other equipment, that can be invasive into the body, they could bring on, some other kind of complications. Aside from just infections in the urogenital region. So you could develop endocarditis, where you basically are having an infection of the heart or the lining of the heart, and that's brought on by the use of medical equipment that contains these biofilms, and it's introduced into the body, and then the body has to fight against it, which causes a lot of inflammation and can be very, very problematic, for somebody who's in a hospital that's already immunocompromised, to develop a disease that threatens the heart tissue. So because we have these complex biofilms, we have a hard time sometimes linking certain microorganisms to a particular effect. According to the authors of your book, it's real difficult for microbial organisms to find their kind of niche in the environment. And here's the picture of a biofilm that's very common to all of us. This is a group of different bacteria that are residing in this person's oral cavity, and it's in the form of plaque. So you see all this plaque residing on this tooth here and on this tooth. It's not just one bacteria, it's a multitude of different bacteria. So there's probably a lot of different Streptococcus alpha hemolytic organisms that are all caught up in this matrix of polysaccharides being secreted by the organisms. And as that plaque sits there, as I said before, the bacteria will start to hunt for those carbohydrates that are brought into the mouth, and with no proper dental hygiene being employed, then those bacteria have a really great environment to start fermenting sugars. And once they start fermenting sugars, they will cause the pH around this tissue to start to decrease, making it more acidic because acid's being produced. And that causes inflammation of the gum, it causes muscle breakdown, tissue degradation, and then eventually it'll also cause cavities to start to form in the actual teeth because it's so highly acidic. So some examples of organisms that help make up biofilms or are associated with them include not just bacteria, but also some fungi, primarily yeast, which we know is a part of our normal flora, so Candida albicans. And then Staphylococcus aureus, which I've already talked about, some Enterococcus species, and then Klebsiella pneumoniae, as well as Pseudomonas aeruginosa. So there's, some different species, and many of these are considered to be, very pathogenic organisms. Pseudoaeruginosa could lead to the development of different eye infections. Klebsiella pneumoniae can affect the tissues in a lot of regions of the body. Respiratory tract tissue gets targeted by this, hence why it's called Klebsiella pneumoniae. And Staphylococcus aureus can also be pathogenic, as we've discussed already. Enterococcus typically can, lead to a lot of gastrointestinal complications. But all these can be pathogenic when given the chance, so they can be considered opportunistic pathogens. But they also reside in these complex structures that we've talked about as biofilms. And that can be very dangerous for us because if we have a biofilm, we don't know what's in it. And with things like these can be very dangerous. Just to give you an idea, Klebsiella pneumoniae, in my laboratories, we don't even work with this organism anymore, primarily because it caused the death of members working at the CDC who are working with it. So it can be potentially very dangerous. And Pseudomonas aeruginosa, many people acquire infections from this particular organism when they decide to bathe in hot tubs that haven't been cleaned properly, or when they can acquire this from flowers that are brought into a hospital, which is one reason many hospitals don't allow you to bring flowers inside of patients' rooms, depending on the immunocompromisation of the patient. Oops, sorry. So biofilms are great for bacteria in order for them to thrive in a community, to share nutrients and resources in terms of supplies, and essentially help for the different microorganisms to survive by working together. They're a problem for us because these biofilms can be very resistant. They can be resistant to antibiotics as well as disinfectants, so we can't clean or treat instruments that are covered with these biofilms because of the resistance issues. When we test antibiotics, we typically test them on Mueller-Hinton agar plates after growing a lawn of bacteria. But for the most part, we are typically just testing antibiotics against one specific organism and not many organisms. The issue that we have with the biofilm is the fact that there's a multitude of organisms that are growing in that film, and we haven't really found an effective way to treat all of those organisms to bypass that film. And what scientists think is happening with this process as to why we're not able to eradicate biofilms by simply targeting them with antibiotics is they think that certain bacteria are exhibiting or expressing different types of proteins that are helping that bacteria to survive inside that community. And typically those proteins aren't expressed when the bacteria is living on its own. So in this sense, you have a very much strength in numbers kind of concept, where different bacteria have different properties. They're expressing different proteins, or they're expressing different defense mechanisms, and because of that, we're unable to touch them if they're hiding in this biofilm structure. So one example we have where biofilms can be present, this might gross you out a little bit, it does to me every time I see this slide, is, this is kind of a microscopic view of a person's contact lens. And contact lens comes into contact with your eye. Some people sleep in their contact lenses, but your contact lenses are very moist. They're not coated with high concentrations of salt, and they're well lubricated. So they provide a great environment for bacteria to reside. And if a bacteria is residing in a biofilm, you can have biofilms that start to colonize your contact lenses. So on here we've got some P. aeruginosa. It looks like that's listed, and you can just see complete colonization all over a person's contact lenses. So this is probably due to a person, not maintaining good eye care. Maybe they're sleeping in their contact lenses, or they're just not taking out their lenses or changing them as regularly as they should. So there's a number of reasons, and it's probably just because you have a lot of buildup of that biofilm over time by not taking care of your contacts. With biofilms, you can have, not just with biofilms, but you can always have, if there's ever an infection or disease or something a patient is succumbing to, it could potentially be more than one pathogen that is inducing that infection. And when this happens, you get a kind of unique attribute of bacteria that reflect a synergistic relationship with one another, which means that both bacteria are playing a different role in eliciting a disease, but they're working together, they're teaming up. We call these synergistic infections. They're also known as polymicrobial infections, which just means you have many microbes working together, or mixed infections. So all of those mean exactly the same thing. You have more than one bacteria that is eliciting the disease. So some examples we have is referred to as ANUG. This stands for acute necrotizing ulcerative gingivitis. So you essentially just have a whole lot of damage done to your gum tissue. You have ulcers, you have degradation of all your tissue. It's hard to recover from something so incredibly invasive. And this can be brought on, again, by a mixture of 2 or more microbial organisms. Other examples include trench mouth, or you can have Vincent's disease, or you can have bacterial vaginosis. So let's kind of talk about the uses of microbiology again, because remember, this is kind of our unit on both our biotechnology as well as our environmental, or as well as the ecology. So we talked about the ecology when we talk about the biofilms. We talk about it when we talk about, where bacteria reside in the body. But let's kind of focus now on some of the biotechnological aspects of it. I guess which also, ties to the ecological portion. But with agriculture microbiology, essentially you have application or the exploitation of microorganisms and playing a significant role in our agriculture development. So we talked in the first lecture about, the different cycles that microbes play a role in, such as that nitrogen cycle, which was the prominent one in helping to provide nutrients to plants in the soil.